Eu tenho que esperar o Eduardo ainda. Just the final arrangement is
Sure.
some people can't come, so we are transmitting your talk for
Sounds good.
people that can't come here. Será que ele já começou? Já? Você começa.
Professor Geert, thank you for accepting to give this seminar. Clarice.
Geert, thank you for coming again to Piracicaba.
True.
Online, but in Piracicaba. Geert has started coming to Piracicaba in 2003. 2003 was his first visit to Piracicaba. And since then, he came to Brazil 10 times, 9 here with us. Only once he went to Londrina and didn't come here. And because of our agreement, 12 of our students went to Belgium for sandwich program, which were very, very successful. Thank you very much for your collaboration with our PhD. And now the word is yours. Thank you.
Thank you very much, Clarice. Thank you very much, all, Fabio and others. It's a great pleasure to be in Piracicaba virtually. But as Clarice was just saying, I have been there enough to picture it very clearly what it looks like. So looking forward, of course, to my next visit. But in the meantime, I would like to talk a little bit about the statistical evaluation of surrogate endpoints in clinical trials, and also a little bit excursions on that indeed. So in a clinical trial, there is usually the interest in assessing whether a certain new medicinal product, a drug, or an intervention, whatever, is working better than placebo or working better than an existing therapy. We usually know what we want to look at in a clinical trial. For example, if we have advanced cancer patients, usually the outcome is survival. But the problem is that often the true endpoint, as I'm going to call it, is rare. In a survival study, whatever the context, you know that if there is censoring, there is no event and there is less information. So also still in a survival study, the endpoint may be distant in time, so it makes studies longer, but also the longer you wait in a study, the more things can happen in between. We call that intercurrent events. So the power of randomization is diminishing as time goes by because all sorts of things happen. And then the idea is that a surrogate endpoint can solve these problems, at least to some extent, by being measured more easily, more quickly, more frequently, not suffering from survival issues, for example. A typical example is the growth of a tumor, and that can be in humans, but of course also in animals. The growth of a tumor growing, staying stable, diminishing, because we can assess that relatively quickly in almost everybody. So that's why we look at surrogate endpoints. Of course, if you replace an endpoint by something else, there is no free lunch, so you will lose some information. The relationship may not be perfect. And I say relationship, not correlation, because it's a bit more complex than just a correlation. There may be other reasons why we do surrogate marker evaluation. For cost, and cost that can be money, but it can also be ethical cost, for example. So in the first few minutes, I will give you an idea of a few examples from various areas. In cardiovascular disease, in myocardial infarction, a heart attack, the true endpoint may be death, but you may look at the ejection fraction or coronary artery patency, for example, instead. Now you see what this might do to a trial, and the number are illustrative. Rather than needing thousands of patients, tens or perhaps hundreds of patients is more than enough. Stroke, the occurrence of stroke is a very serious thing, but you may need to wait for a very long time. You may need to enroll large numbers of people. But if you look at blood pressure, for example, you will get the information more quickly and on a much smaller sample size. Of course, you may say immediately, but yeah, blood pressure, diastolic blood pressure, is that now a good replacement for stroke? We may doubt that a little bit for good reason. So we need evaluation. We need to evaluate whether a replacement endpoint, a surrogate endpoint, is valid. So in terms of terminology, the true endpoint or clinical endpoint is the endpoint on which you would like to have an impact. A biomarker in between, I talked about surrogate endpoint, not yet about a biomarker, but a biomarker is a usually biological measurement that you can take in a trial, but not necessarily in a trial, in healthy circumstances, in disease circumstances. So is that everything? No, for example, a scale. If you have a questionnaire that leads to a scale, cognitive functioning, mental well-being, quality of life, that's not a biomarker, but it can still be used as a surrogate marker. A surrogate endpoint is very precise, whatever endpoint, a biomarker or actually something else that you can use instead of the clinical endpoint with good advantages and not too many disadvantages. That's a way to put it. Few other examples still from oncology. Ovarian cancer. The CA125 gene marker is a very important predictor for the overall survival, for example. I will not read all of those, but just a few. For example, in prostate cancer, prostate-specific antigen and its evolution over time may be a very important marker for time to progression, overall survival, etc. There are more. I'll pick out a few others. Osteoporosis. The true endpoint is, of course, fracture. As we get older, bones get a little weaker. Fractures are unfortunately more likely to occur. So bone mineral density, I said it, may well be the thing you want to look at. Cardiovascular ejection fraction for myocardial infarction, for example. HIV, I'll come back to that. There was a time when the outcome was AIDS, and in some cases, time to AIDS, and that means the clinical diagnosis of AIDS, or even more so, time to death. Came a time that people started measuring CD4 counts. Later, viral load. Later even, whether viral load, and that's what we do now, viral load is below the detection limit, yes or no. Ophthalmology in older people that are progressively deteriorating in vision, visual acuity can be used as the true endpoint, is often used as the true endpoint, but intraocular pressure, so the pressure, the liquid pressure in the eye, can be used as a surrogate, for example, and so on. An area in which we have been working, and that's why you have many examples here, is Alzheimer. Now, the diagnosis of Alzheimer is a little bit more complicated than, for example, in advanced cancer, where we look at death. So what would be the true endpoint. Often it's a cognitive scale. The one I'm mentioning here is called CDR, sum of boxes. So CDR is cognitive dementia rating, sum of boxes. You have other scales, the ADAS-Cog, the mini mental state examination, for example. So even with the true endpoint, if we say it's cognition, how do we measure cognition that's not without ambiguity? Surrogates, more and more do we look at various biological markers with brain scans, for example, like amyloid plaques, like tau PET scans, etc. One cognitive scale can be a surrogate for another cognitive scale. And of course, that's the first time that I'm going to say that here. Sometimes it's useful to combine various endpoints. If one endpoint gives an imperfect view, maybe we should look at it multivariately. Amyloid and tau together, for example. Okay, just switching to another endpoint may actually be a little shaky. There is a very infamous bad precedent in the so-called CAST trials, the cardiac arrhythmia suppression trials. So people with arrhythmias take medication to keep their heart rhythm under control. And the drugs, and this is an old study, we go back to the late 80s, early 90s, encanite and flecainide were working very well. They really reduced arrhythmias. The problem was that actually people who took the drug had a strongly increased death rate from heart attacks. Now, that's a bit strange, because you have a drug that works in the cardiovascular atmosphere. It works well, but then it creates a side effect, and a very serious one, because people died also in the cardiovascular sphere. How can that be? Well, for example, if arrhythmias are controlled too much, the heart will basically always beat at the same rate. And that's not a good thing, because it should basically adapt itself to your level activity. So level of activity. Suppose you're listening to a statistical presentation like you are doing now, and you think it's boring and you almost fall asleep. You don't need a lot of energy. But if you're very active and you go to a dancing party tonight, then you need a bit more energy, and your heart actually should adjust to that. Those people who do sports know that very well. The heart has to adapt. So if the heartbeat is controlled too much, you actually may be overdoing it. So you need to be very careful, of course. So a bit of notation. I try to keep at least the first part of the presentation notation low. We have three players. We have three things that we have to manipulate. The treatment, and for the sake of argument, I'm going to keep the treatment binary. Let's say control and experimental. Then I have a true endpoint, and if all goes well, the surrogate is sitting in the causal chain from the treatment to the true endpoint. For example, you may say the treatment is diet, standard diet versus healthy diet, modified diet. Colorectal polyps may be your surrogate on the way to colorectal cancer. And that seems very logical, but you may have not only hyperproliferation, you may also have apoptosis. Two different mechanisms that provide two pathways from treatment to true endpoint. If that's the case, not all the information flows through the surrogate. It's like you have a river, and the river has two branches that go out and then come back again. Not all the water flows through a single branch. And then the surrogate is not able to completely capture the information of the true endpoint. Honestly, this is almost always the case. Okay, let me give a simple example from ophthalmology. We have people with the ophthalmological trial that we are considering here, the ARMD trial, age-related macular degeneration. So people's vision decreases with age. And in this study, they are given a drug or placebo. It's an old study, 28 years old. So they get interferon alpha or placebo. And the outcome, the true endpoint, is visual acuity, the number of letters you can read on a vision chart. Those of us who have their eyes checked regularly know that you need to be able to read letters, big ones to begin with, and then gradually smaller letters. And the surrogate, and that's also perfectly possible, is the same thing, but then 6 months earlier. It's a bit like an interim analysis on another endpoint that is actually very related to the true endpoint. So we look at change versus baseline. That's why every patient can have both positive and negative values, as you see in the graph. And this is one of those vision charts that this is not a standardized vision charts. They typically have 72 letters, but you get a sense of what I mean by that. Okay. How do we evaluate a surrogate? Well, what I would like to do with you is build it up gradually, step by step, and as like a building. And a building starts on the ground floor. So the ground floor is the definition of a surrogate as produced by Prentice in 1989. Prentice said, well, a good surrogate would mean that if you test the effect of treatment on the surrogate or on the true endpoint, it's equivalent. So testing the null hypothesis of no treatment effect on the surrogate and on the true endpoint are equivalent. That's very intuitively logical. But what does it mean in practice? It's very hard to think of what is equivalent here. What do we mean by that? Does it always have to agree? But we only have one trial. And if there is no treatment effect on the surrogate and no treatment effect on the true endpoint, is that evidence or is that maybe just lack of power? So we need to think about that. So Prentice realized that and he came up with criteria. S, T, and Z are my variables. So as a reminder, S is the surrogate, T is the true endpoint, Z is the treatment allocation. And if you have three variables, you can construct three pairwise models out of them. So we have two treatment effects. We have the treatment effect on the surrogate alpha. We have the treatment effect on the true endpoint, and we have the direct relationship between the surrogate and the true endpoint. So this looks like three very simple regression models in the case of continuous data. Of course, if you have binary data, count data, you would need to use a generalized linear model. But of course, everybody in Piracicaba knows all about generalized linear models. I'm just illustrating it for now with linear models indeed. And there's one additional thing. We allow the regression on S and on T to be related with each other. That's why we have a covariance matrix. Okay. So Prentice's definition, again, but the criteria in words is treatment is significant on the surrogate, treatment is significant on the true endpoint, and the surrogate itself is directly significant on the true endpoint. But indirectly, the treatment is not significant on the true endpoint. So indirectly, by that we mean conditional on the surrogate, treatment is not significant on the true endpoint. These criteria, again, look intuitive, but this is the first floor of the building. There's a problem with it. The definition and the criteria, the four criteria here are not equivalent. And that's a bit of a problem, of course. Moreover, requesting significance, well, it may well be that you have, let's say, several trials, and in some of the trials, there is no significance at all. That's not necessarily against surrogacy, because if surrogacy works, yeah, they would be equivalent, and that means not significant would also agree with not significant, and Prentice did not allow for that. This is the same thing, yet formulated in a different way. We have the two treatment effects, beta and alpha, unadjusted of the treatment on the true endpoint and on the surrogate. The third criterion with parameter gamma is the direct relationship between surrogate and true endpoint. And then the fourth criterion, sometimes called the criterion, says that there is no treatment effect on the true endpoint if you take the surrogate into your model. Okay, Larry Friedman and colleagues, they actually realized this, and they went to the second floor of the building, or they built the second floor of the building by saying, you know what, we will, rather than having these criteria, which are of a hypothesis testing nature, let us quantify. And it's good to know that Friedman, Grobart, and Shatskin came from an epidemiological background. So in epidemiology, we have a quantity called attributable fraction. How much of the effect can be attributed to a certain cause? So he came up with a proportion explained, saying that if you have, let's say, your two treatment effects, unadjusted beta, adjusted beta S, from that we can construct the so-called proportion explained. And it looks intuitive at first sight, because if beta S is equal to 0, it means that the criterion number 4 is completely fulfilled, but then the proportion explained would be 1. If, on the other hand, conditioning on the surrogate would have no effect whatsoever, then beta and beta S would be identical, but then the proportion explained would be equal to 0. Sounds like a nice story, but unfortunately, it's not entirely correct. We have shown, many people have shown, that the proportion explained is not even a proportion. It can be negative, it can be greater than 1. It depends on nuisance parameters in a way that you don't want, so it's not a good quantity. It's a nice concept, but it's not properly quantified in this way. So in 1998, two people, if I'm not mistaken, Mark Boyce and myself, proposed two other quantities. Now you will see that one of those quantities is in red and the other one is in green. The relative effect is the ratio of the two treatment effects. And if all goes well, and if that ratio would be constant from one trial to the next, then if you would know the relative effect and alpha, then you would know beta. You can simply solve for it. But of course, the problem is we are not entirely sure that the relationship between those two treatment effects is not only linear, but actually multiplicative. That's a very strong condition. So that's why we need to amend that a little bit. We need to think about it a little more. The problem is that it's hard to know whether this is a meaningful quantity from a single trial only. On the other hand, these authors, so Mark Boyce and me, produced the adjusted association, which is the correlation at the individual level between the surrogate and the true endpoint, given the treatment arm one belongs to. And that's in green because usually that measure behaves rather well. So let's apply it to the ARMD trial. You will see that criterion 1 and criterion 2 are not significant. So in parenthesis framework, it would be game over. Of course, we go to Friedman and the proportion explained. Yeah, it is a number between 0 and 1. So far, so good. But look at the confidence interval. That's another problem. It often has a very wide confidence interval. By the way, the relative effect also. So I think from all what we have been doing, definition ground floor, criteria first floor, proportion explained second floor, relative effect and adjusted association third floor, we still need to go one floor up. One thing we take up is the adjusted association because we find a correlation of 0.75. That's not too bad, but moreover, it is very precisely estimated because, of course, we do not have replication of the treatment effect in a single trial. It is what it is, but we have replication at the patient level. Okay, so in summary, the proportion explained is not well defined, it's scale dependent, it depends on nuisance factors, it's very wide limits. The relative effect has potential as a concept, but it really doesn't work well on a single trial. So that's why we and others have decided, why then don't we look at multiple trials? we think of meta-analyses, or if that's not possible, maybe a multi-center trial where we use the centers as different units, or even several meta-analyses. Okay, so the relative effect is now going to be brought very soon, on one floor up, to what we call trial level surrogacy, and the adjusted association we will keep more or less, but that will become the individual level surrogacy. Albert in 1998, and that's already 27 years ago, considered this a good idea to use alternative approaches, but also considered it not a good idea to use the proportion explained. So what we are going to do now is going to meta-analysis. It can be multiple centers within a trial. It can be multiple trials within a meta-analysis, but let's just call it meta-analysis. So we need to expand the notation a little bit. Zij is the treatment assignment, 0 or 1, for patient J within trial I. That's why I was using before the index J. Usually we use I, but now I can still say J is patient J, and I stands for the trial. Tij, Sij, similarly. And of course, we are going to look at the treatment effect in every one of the trials separately. So we have a treatment effect for every trial I, beta I, alpha I. The trial level surrogacy, that's going to be the relationship in the pair alpha I, beta I, and the individual level surrogacy, that is the relationship in the outcomes, the two outcomes, surrogate and true endpoint, for given patients. So these two need to be kept apart. There's statistically two different things. We notice that in the clinical world, people are often seeing very high individual level surrogacy and then decide it works at the trial level, but no, you need to do both validations separately. This has been developed over the years by us and other people for all sorts of outcomes. Gaussian, binary, time to event. Also for settings where the surrogate and the true endpoint is of a different type. For example, your surrogate may be tumor growth. Does it grow? Does it shrink? Does it remain the same? That's a categorical variable. But the true endpoint may be survival. So you need to have models that allow for different data types in its two outcomes. Of course, I already referred to that very briefly. When we say prostate-specific antigen, we may measure that repeatedly over time. And that repeated measures profile can really have very nice additional information over and above a single measurement. So we need to be able to take that into account, etc. And then, of course, like I was hinting on in Alzheimer, you may want to look at amyloid plaques and tau PET scans at the same time. So more than one surrogate, because together we know more than each of us individually. Okay, so what did we do? We used, you can guess, linear mixed models where you have two outcomes per patient, but you also have repetition of the trials. The nice thing is that we can take it one step further methodologically. So Prentice and we have produced, starting with Prentice. Prentice has done hypothesis testing. That's the first methodological layer. Friedman and us, we have produced estimation methods, estimate quantities, measures that we propose, and we still do. But of course, if you have several trials, you can also look at the next level, which is no longer hypothesis testing only or estimation only, it's prediction. What would happen in a new trial? So suppose I have done the validation, it works. I now have a new trial. I would then evaluate in the new trial only the surrogate and predict from there the effect on the true endpoint. You can do that at the individual level, but first we do it at the trial level. By the way, what we can do in this case also is still quantify. Now, quantify the trial level surrogacy by a measure that we can all interpret very nicely, and that is calibrated between 0 and 1, the R-square trial. It's an R-square measure. So for ARMD, 0.70, which is not super, but it's not bad at all. At the individual level, the R-square individual is 0.48. It's a little lower. Of course, the R-square, if you take the square root of it, you get R, 0.69, which is not very far away from the adjusted association that we had before. So this is a case in point that the adjusted association and the individual level surrogacy are very closely related. Only now we evaluate it in a meta-analysis. Very briefly, a number of other case studies. We have the first column, age-related macular degeneration, and then we have two cancer cases, ovarian cancer and colorectal cancer. You see the endpoints. I'll focus on the cancer cases for now. Progression-free survival as a surrogate for overall survival. Now, if you look at the Prentice criteria, it's not working all that well, but neither is the proportion explained. Look at it. The proportion explained in the ovarian case, 1.34. There we go. It's bigger than 1. Shouldn't be the case for a proportion. Case in point, it is ill-defined. In the colorectal case, 0.51, but look at the interval. The relative effect, by the way, which we do not promote, is also having very wide confidence intervals, impossibly wide confidence intervals in the colorectal case, whereas the adjusted association is not too bad. But look at the R-squared trial measures, 0.94 at the R-squared trial, 0.94 in the ovarian case, 0.57 in the colorectal case. Similarly, at the individual level, 0.90 in the ovarian case, 0.57. By coincidence, they're the same. They don't have to be in the colorectal case. Now, if I tell you that in one of those two cancer cases, exercises like this have led people to use progression-free survival rather than overall survival because the meta-analytic or multiple unit validation worked well, maybe some of you will say, oh, I know, it will be the ovarian cancer case. No, it's not true. It's the advanced colorectal cancer case. Yeah, but the R-squared trial is very high in the ovarian case. Yeah, but the only problem is that progression-free survival in ovarian cancer happens so close to overall survival that the time gain is too limited to change. Colorectal cancer case, 0.57 is maybe not great, but don't forget it's an R-square. The actual correlation underlying would be a bit higher. And there is an enormous time gain. So there are many colorectal cancer trials now where the outcome is simply progression-free survival. So the surrogate has become the new true endpoint. Another case in schizophrenia, we have two data sets, one that can be split into units in two different ways. Investigators, 138, or principal investigators, 29. And then there is a second study. The Prentice criteria, I can be brief about it, they don't work. And look at the proportion explained now. We now have a case, we've seen it all. It's negative, and the confidence interval is the entire real line. Pretty useless, same with the relative effect. The two adjusted associations, they always stand out a little bit among the single unit validation measures. They're usually stable and precise, 0.72, 0.74, with a very narrow confidence interval. If you look at the R-squares and you summarize a bit, it's the R-squared trials between 0.60 and 0.70, the R-square individual, 0.50, 0.55. So we get a coherent picture in this way. Okay. Now, there is one thing I have to confess. When we are analyzing the ovarian and the colorectal cancer cases here, we're actually treating the outcome as Gaussian, continuous. And now you may say, hey, wait a minute, that's not the way we analyze survival data. I agree. So here is the result in the ovarian cancer case if we would use instead survival models with copulas. I will save the details, the Clayton copula, the Hugard copula. And the bottom line is that at the trial level, we formally still have an R-square. For now, I have to say at the individual level, yeah, we have a different association measure, but fortunately, we can formulate that as Kendall-Stau. And Kendall-Stau also is bounded by 1, so we have a meaningful interpretation also in this case. And actually, both in ovarian and colorectal, a fairly similar picture of what we have before. Okay. Let me now go into a few extensions. For example, when I have 2 longitudinal endpoints, that means the surrogate is measured longitudinally over time, the true endpoint is measured longitudinally over time, and maybe I want to use the entire longitudinal surrogacy sequence for the entire true endpoint. Or maybe I want to use a part of the surrogate sequence, the early part, because the less measurements I take from a sequence, the quicker I can do my analysis and the more time I gain, but the less information I will have. So there is that trade-off, of course. All right, without going into detail, together with Ariel Alonso, we have developed a number of measures. The variance reduction factor, the likelihood reduction factor, R² lambda, and a few in between. Now, maybe some of you may say, R² lambda with a capital lambda, that reminds me of Wilks lambda. Yeah, for a good reason. This measure for continuous cases is very strongly related to Wilks lambda. The theta p is related to Pillai's trace, for example. So they're related to the classical root statistics as we know them from multivariate analysis. Okay, but we developed one more, and that is an information theoretic measure. And here I need to go out of the usual comfort of the usual precision measure that we used. What do we mean by information theory? So by information theory, we're actually trying to circumvent a well-known problem with the exponential family, and therefore with the generalized linear model. No matter how much many people like the generalized linear model, the mean variance relationship. We like to quantify the uncertainty with a fairly stable measure here, and information theory does provide that to us. I will save us the details, but just say that if you look at the expression without going into the details, the R²H, which is our information theory-based R², and the proportion explained look very similar in spirit. EP stands for power entropy. PE stands for proportion explained, sorry about that. Power entropy is a matter of, let's say, divergence, variation, but it's a different quantification of variation than, for example, the variance. It's a competitor, or rather an alternative to the variance. And of course, we look at the, let's say, I'll keep calling it variation, but it's actually, or entropy, in the treatment effects given the surrogate treatment effect or not given the surrogate effect. And of course, you hope that the same applies that we were hoping with the proportion explained, namely that it's a calibrated measure. It is. The R²H is a calibrated measure. 0 means no surrogacy whatsoever. 1 means perfect prediction, perfect predictive power. Not the same, but perfect prediction. Prediction can follow a deterministic function, and that would be fine, of course. Okay. So in the schizophrenia trials that we had before, using continuous outcomes, the variance reduction factor, one of the measures, would be 0.40. By the way, I forgot to say, variance reduction factor, the R² lambda, they all lie between 0 and 1. You see that's very precisely estimated. The R² trial, 0.85. So we get pretty good results in this case. For binary outcomes, you can dichotomize your outcomes and see what it does in the binary case. Again, we have trial level R² and individual level R², namely the R²H or the R²H max. Let me say a word about the R²H max. I've been saying several times that the R²s lie between 0 and 1 as they're supposed to be. True, but in the binary case, R square H lies between 0 and 1, but cannot reach 1. There is an upper bound below 1. So we recalibrate it, make certain with the R square H max that it can reach 1 if it would be perfect. Otherwise, it's difficult to interpret. One more, age-related macular degeneration, both outcomes binary, R square trial at the trial level, 0.38, R square H max, which is at the individual level, 0.50. You start to see it's more of the same, and you start to feel also how you can interpret it. Of course, always against the background, what gain you will have also in terms of time and ethical cost, etc. Okay, how do we proceed, for example, in survival data? Suppose the surrogate endpoint is time to progression or death, so progression-free survival. T would be overall survival. You fit a Cox model with and a Cox model without a surrogate, and you calculate based on that your R-square measures, R-square trial or R-square individual, R-square H at the individual level. And you see, we actually get pretty decent results at 0.80, 0.85 in almost all cases. So pretty strong in this case. Okay. I talked about prediction in a new trial. So here is the ideal world. You do the evaluation of a surrogate endpoint. It turns out to be fantastic. Okay, then we can say in the next trial, we're not going to look at the true endpoint anymore. No, we will only look at the surrogate. And then once we have the surrogate, we will predict the treatment effect on the true endpoint using the prediction rule that I derived from my evaluation exercise, including confidence intervals, p-values, and everything. That's the ideal world, which does not exist. Because there are three factors that determine the precision, the uncertainty, if you wish, on such a predictive exercise. The sample size of the new trial, the sample size of the meta-analysis, because that's not going to be infinitely large, and then actually the R-squared trial as such. Three sources of variability on top of each other. It means that using it for prediction in the way that I have proposed in practice is not going to happen. That's not the way the FDA uses it. That's not the way the European Medicines Agency uses it or other regulatory offices. Rather, the exercise is interpreted in the boardroom. The value is high. There's a lot of time gain. So we consider that a surrogate, and the FDA actually has a taxonomy now, like reasonably likely surrogate, etc. The use of a surrogate is not necessarily always replacing full stop. You can use a surrogate for provisional approval. You can bring a drug or something else on the market while you conduct further studies. You can do that based on this. Let's say you have to bring a vaccine on the market very quickly. You can do that on immunology while you keep evaluating the infections, and then maybe later you confirm or don't confirm the marketing. Of course, this has to be done when it's sufficiently safe to do that, but that's another discussion. So this means we have to reflect on the role of surrogates, but I think I just did that. Okay, in the remaining minutes, the remaining time, I'm going to talk a little bit about yet another framework that we have proposed, and that's gaining quite a bit of use these days, and that's the one of potential outcomes. And I'll work in two steps. It's work also with Ariel and with Wim van der Elst. So Rubin, when he presented the Rubin causal model set, when in a trial, and that's true for a clinical trial, but also for an agricultural experiment, when nothing is missing, actually half of it is missing. Because think of a patient who is assigned to control and active, then the same patient, to control and active, then you would have the true endpoint under control and the true endpoint under active. Of course, that's not possible unless you have specific designs or crossover trials or something like that. But you get only one of the two. That's why we talk about potential outcomes. There are two outcomes existing, only one is observable at the same time. What we are interested in is the individual causal effect, the ICA. What is the treatment effect if I take somebody's true endpoint under experimental minus the true endpoint under control? And the expectation of that is the treatment effect. And then the question is, can the surrogate predict that beta? In a classical clinical trial, this problem also occurs. In an agricultural experiment, this problem also occurs. Why don't we worry about it? And I'm saying that because I mean it, not just to please Clarice. Good experimental design. Through good experimental design, we can compare groups and actually get access to that data, which is otherwise inaccessible due to proper randomized design. Okay, we have the predictive causal association, but there is a bit of a problem. All of this now depends on the joint observation of T0 and T1, which we can't. So what do people in causal inference often do? Make assumptions until it's identifiable. We refuse to do that, and we say, we're not going to make assumptions, we're going to do a sensitivity analysis. So let us range over all possible values of whatever is unidentified, and then construct the individual causal association distribution. Bayesians would call it a posterior distribution, and in a way, it's related. It's not exactly the same. And in a case like this, I would say the conclusion is clear. And it's not very good because I'm very close to zero. Nothing is happening in this case, for example. Okay. Now, you may have noticed that I have two potential outcomes for T and only one outcome for the surrogate. And that's a little bit asymmetric. So the full causal model says, yeah, we have the two endpoints under control, and we have the two endpoints under treatment. So then we have 4 potential outcomes. Every patient contributes only 2. Okay, so then the question is the individual effect on the surrogate, delta S, predictive for the individual effect on the true endpoint, delta T? The non-identifiability is there even stronger because I have 4 outcomes in mind, but only 2 in my data set. Okay, so we have the treatment effects. We can build a model for the 4 outcomes. For example, with continuous data, a 4-dimensional multivariate normal, a quadrivariate normal, and that, of course, projects onto a bivariate normal using the classical rules on the normal distribution. Okay, the individual causal association as it is now is the correlation between the individual causal effect on the true endpoint and that on the surrogate endpoint. We have a 4x4 correlation matrix. Not every correlation is estimable from the data. So we do a sensitivity analysis, and here comes, we now have to realize that in general, for everything you do, information comes from 3 sources. Design, good experimental design, data. Design and data are good things. Assumptions, yeah, if we have to, but if we can avoid them, it's better, of course. If your design can be very clever so that you can reduce the assumptions to things that are a bit more plausible, why not? But if I have to make, I can actually make assumptions. You know that I'm not a great fan of making too many assumptions. We do sensitivity analysis then instead. So here is an example. In the case of the age-related macular degeneration trial, we have this new measure now. At the bottom, you see the histogram. So if you take a very negative attitude, you say, everything between 0 and 1 is possible. True, but that's not the way statisticians think. We say the mass is really shifted to the right. So here I have a feeling that we have a pretty decent surrogate. You could calculate the average or the median, and you would see you get very close to the measures that we had before. So moving towards the conclusion, it doesn't stop here because as I was already saying, in the context of Alzheimer, but not only there, I can look at a whole collection of surrogates. That can be a single outcome measured repeatedly over time. It can be several different surrogate variables simultaneously. For example, if in Alzheimer the true outcome is CDR sum of boxes, one cognitive scale, I can think of the surrogate as amyloid plaques, tau PET scans, 2, the ADAS-Cog scale, 3, the mini-mental state examination, 4, and now to really open it up, I can measure all of that longitudinally. That's a wealth of information. Of course, it takes a lot of effort to collect all that, but believe it or not, that is often done in clinical trials. And not only that, we have that information also in registries. So, of course, the models will expand. Mathematically, it's just more of the same. You get P+1 outcomes that are measured or that can be measured under 2 conditions. So a big multivariate normal that produces a measure of association. Now, one thing that I also would like to mention, and this is a bit more practical, suppose that I want to bring data together in a meta-analysis. In clinical trials, that can be, let me stick with Alzheimer. Similar trials in Alzheimer, but by different companies. Roche, GSK, Johnson & Johnson, what have you. All sorts, Biogen, all sorts of companies. Will they be happy to share their data? I don't think so. You can, of course, translate that also to commercial products in the agricultural sphere, for example, or in many other commercial circuits. People are a bit protective of their data because of commercial reasons. So we have actually proposed the use of a federated data analysis method where we don't get the data centrally. No, we send out to the various sponsors our code, for example, and then each one analyzes their data. We make certain that they standardize the data in the data set in a way we want. Then they do the analysis, and what we will get is parameter estimates and variance-covariance matrices. You cannot reconstruct the data from that, and then a central combination is applied. Of course, what does it mean? Your method needs to be two-stage. If the only way of doing it is one single model, it's not going to work. But if you can also do a meta-analysis by analyzing every trial separately and then bring it together in one big thing. And that's what we have been doing, for example. So that's the workflow that we have been doing, for example, in the case of Alzheimer. Of course, it takes a while before people buy in. So the steps that we took is we analyzed fake data, simulated data first. Then we also analyzed, and I did not mention that yet too much, natural history data, registry data. Now we're currently working on the analysis of real trials, but only the placebo arms. Of course, by definition, that doesn't give any treatment effects, but it shows how the analysis is done, and we can get a good idea about the individual level surrogacy already. And soon it will be applied to also the active arms. I'm giving an example of Alzheimer because that's what we're actually doing right now. It's also going to be done in Parkinson, and it can be done in many other settings as well. Of course, I discussed already that there are several issues to be solved, and that's true in every other area. You change it to your own problems, and you will see that very similar problems occur. Like what measures are you taking as even the true endpoint? If you have PET scans, are they standardized between the various equipments? I'm sure several of you in whatever field you work may say, yeah, we analyze the data, but if they come from different sources, standardization may be a bit of an issue, etc. We have measurement error models for that, but more work needs to be done as well. Okay, natural history data can be analyzed too, as I said. So here's an example from APNI. APNI is an international initiative on Alzheimer's disease, which is freely accessible to anybody who wants to analyze the data. You can have longitudinal markers of all sorts of things. So here is an example of the analysis of natural history data, where we look at the patient level. Let's look only at so the VRF again is the variance reduction factor. That's one of the R-square measures on that database. I will concentrate on that one. So, okay, you see that there are actually four sub-analyses. CN stands for cognitively normal, people who do not have dementia. MCI is mild cognitive impairments. People function okay, but you see that the cognition is no longer exactly as it should be, and then you have full dementia. You can analyze them all together or in subgroups, and then you see that the VRF is actually, yeah, okay, but not great. Something that I'm not showing yet, but we have done in the meantime is if you combine the outcome here that we analyzed indeed was tau PET scans, but if you combine that with amyloid plaques in the brain, you get a much better R-square. It goes to 0.40 or even a bit higher. Okay, so in conclusion, in view of the time also, are surrogates useful in practice? Like so many things in life and in science, the answer is not yes or no. It's not black and white. You use it or you don't. But it actually can enhance, speed up, improve quite a few things in clinical research, but actually in a lot of experimental context indeed. There is individual level, there is trial level, any more complex hierarchies, there are more levels. I always say some of the colleagues working on the field use the term use the word surrogate marker validation. Well, I prefer the word evaluation. It's more modest. We statisticians, we evaluate and we present the results, we explain it, but there are many other factors that need to be taken into account before a surrogate is actually used in practice. It's regulators that have a say on it. And I think very important in human studies, you need also to have a conversation with the sponsors, the companies, or the academic consortia or whatever, but also with the patients and their representatives, because they are also a very important player. And more do we see also in Europe that patient organizations are actively part of evaluation exercises. A surrogate can be evaluated and can be considered good for now. But if we have a drastic change in therapy, for example, in cancer, for a long time we had cytotoxic therapies. Now there is also quite a bit of immunotherapy. Well, if you have a change like that, it doesn't automatically mean that your surrogate is still valid. You may have to reevaluate, of course, and then you have a new class within which you can apply it again. So it's not a silver bullet, it's not blue sky, but I think we can usefully contribute. I feel we have usefully contributed to clinical and other experimental sciences. On that note, thanks again for the invitation. Thanks for your attention. I will stop here and very happy to take questions, of course. The word is back to you in Piracicaba.
Thank you, Professor Geert, for presentation.
My pleasure.
Now we will take a question.
Alguém quer perguntar alguma coisa? Well, Gert, this is a very new topic for our students because most of them don't work with the health data in general.
Yes.
Work it, but it was very interesting, and I am sure that we can have ideas for some agricultural data from your talk. I'm sure.
Yeah. On that note, Clarice, I actually I hold the opinion as past president of the IBS, which you also are, that it's a very good idea to try and foster cross collaboration between subfields of statistics. So in that sense, I think the agricultural biostatistics and the clinical biostatistics should keep talking to each other all the time. So therefore, I think that at one point we can discuss it a little further and see how certain concepts are very specific to one area and the other, but knowing what's happening in the other areas often gives ideas. It's cross-fertilization, if you want to call it that way, serendipity, symbiosis, all of those terms apply not just to biology, but also to our sciences.
Yes. Thank you very much, Geert. And I hope to see you soon.
Yes, and likewise, take good care. Enjoy summer coming in Piracicaba. Here in Belgium, it's evening and winter is coming, but it is what it is. So thanks again for the invitation. See you guys in the near future.
Thank you.
Thank you.
Bye-bye, Geert.
Bye-bye.
Bye. Oh, não deu tempo.
Agora foi.
Ele saiu.